In this final video on concepts related to Chapter 32, covering details about general characteristics of animals, animal body plans, the development of animals from an embryonic point of view, and also body cavities and other developmental trends, I want to show you how all of that information can be put together in the assembling of phylogenetic trees of animals. And so there are two ways we can look at animal phylogenetic trees. And one of the emphases can be made on the morphological classification. A morphological classification of animals is one that deals with the forms we can recognize not only as adults, but also throughout development. I'm talking about embryonic development. And so let's take a look at this phylogenetic tree on this slide and begin making a few observations. Number one, this tree is going to be a rooted tree because it identifies what is hypothesized to be the ancestor to all animals. And that is going to be some form of a colonial choanoflagellate. These animals live in colonies, meaning that it's just a group of many single-cell organisms. From here, we go to metazoans. So this is going to be a large clade of organisms. All of the animals are metazoans, and it simply means that the body is made up of many cells. Truly multicellular organisms, in this case, members of the animal kingdom, are going to belong to the clade metazoa. And so early on in this history, or this hypothetical tree for the classification of animals, one group remains pretty much unchanged, and that is going to be the phylum Porifera. The sponges are considered to be not only the basal animals in this case, but they're also sister taxa. So if you look at this tree, the sponges are going to be right there, sharing a common ancestor with the eumetazoans. The rest of the animals in this phylogenetic tree are going to be animals with true tissues. As we've explained before in class, true tissues means that the cells have certain features connecting them one to one another and ways in which cells can collaborate with one another. Now, when we go to the eumetazoans, animals with true tissues, we are going to make another inference, and that most of the animals are going to belong to a clade known as bilateria. As you can imagine, bilateria makes reference to the bilateral symmetry for the bodies of these animals. So what is multicellular but doesn't have bilateral symmetry? Oh, it's going to be animals like the cnidarians, jellyfish, anemones. Those are going to be members of phylum Cnidaria, and also the comb jellies. The comb jellies are described as having biradial symmetry. So they have true tissues, but they don't have bilateral symmetry, so we leave them outside in another couple of other groups. But if we focus on the bilaterians, we're going to see that one way in which animals can be further subdivided is going to be based on developmental trends, like when the mouth forms. And you learned this in a previous video and also from your reading that deuterostomes are going to be animals where the mouth forms second. There are, of course, other characteristics that are typical of deuterostomes. We mentioned, for example, for deuterostomes, they have something called a radial cleavage, that the cells in an embryo don't have a predetermined structure they are going to become early in development. You can remove a cell and it's going to go on to become a complete adult individual. Also about deuterostomes, you learn that the coelom is going to be forming from pockets that emerge from the gut of the animal, a primitive gut, which is known as the archenteron. And then we also have the protostomes. These protostomes are going to be those where the mouth forms first. the blastopore of the two-layer gastrula goes on to become the mouth of the animal. Mouth first, protostome. And so here we see all of those groups of organisms that can be classified as protostomes. Another detail that I want you to notice is that everything here in the clade bilateria are going to be animals that are triploblastic, meaning that they have 3 germ layers. So this is going to be a way in which we can form a phylogenetic trees for the animals. And it's based mostly on characteristics you see in the body of the animal and also during embryonic development. However, scientists have rearranged a little bit how animals are classified and proposed more of a molecular phylogenetic tree of animals. Notice that some of the trends still carry over from the previous phylogenetic tree. For example, we still have the colonial choanoflagellates as the ancestor to all of the animals. The sponges are the outgroup, but they're still a sister taxa to all of the eumetazoans, animals with true tissues. From here, the eumetazoans, we can subdivide all animals into those that have bilateral symmetry. We have the clade bilateria. And then we have the cnidarians and ctenophores separate in another group. When we go to the bilaterians, we still have a clade known as deuterostomia, the deuterostomes, animals where the mouth forms second. But instead of protostomes, we're going to see 2 subdivisions, the Lophotrochozoa and the Ecdysozoa. And these 2 are going to be subdivisions based on molecular differences. The kinds of genes that control the development of the embryos are going to allow, for example, for these Lophotrochozoans to form structures like, I will take the time to write these. These Lophotrochozoans are going to be those where the embryos sometimes show a feeding structure known as the lophophore. It's a mouth that is surrounded by tentacles for aiming, guiding food in the direction of the mouth. Among these members of the clade Lophotrochozoa, we're also going to be finding many animals here that form an a larval stage known as the trochophore. And so those 2 features are going to be present consistently among members of this clade Lophotrochozoa. And the other clade we have for animals that have bilateral symmetry but are not deuterostomes are going to be the Ecdysozoans. And these Ecdysozoans, we can see 2 main clades, nematodes and arthropods. And what they do is that during growth and development, as they go from embryonic stages to larval stages, juvenile stages, adult stages, they practice something called ecdysis. Some people pronounce it also ecdysis. This ecdysis is going to be the molting of an old covering of the body. Imagine like an insect capable of molting the exoskeleton so that it can continue growing. Before they can get rid of the old covering, they have to begin making a new covering from inside of the old skin. So when they break out of the old exoskeleton and shed or molt that exoskeleton, there is a new one that is already in the process of being formed. It may take a couple of days to completely harden. That process of shedding the old covering and making a new one is what we call ecdysis. And that is going to be characteristic, as I said, of these 2 main groups. There's going to be other animals that are also classified as ecdysozoans, but the 2 main ones we're going to be studying in class are going to be the nematodes and the arthropods. So this is going to be the 2 main kinds of phylogenetic representations for the animal kingdom we're going to see. And in class, we're going to focus primarily on this molecular phylogeny of animals as we continue exploring more about animal diversity. that is all I have to share with you for chapter 32. It's time to study and review, and remember to write in detailed explanations for the objectives in this class. Goodbye.